Okay, Dr. Rolando, now we are going to start.
Perfect.
Okay, so good morning and welcome you all in the ongoing webinar series of Mizoram University. First of all, I would like to thank our Honorable Vice Chancellor, Professor K.R. Sambarasiva to initiate this webinar series during this pandemic situation. And in this line, today we have a Dr. Rolando Gittens, scientist from Institute for Scientific Research and High Technology Services, Panama. So before we begin our talk, I would like to give a brief introduction of Dr. Rolando. So Dr. Rolando received his bachelor in electrical and electronics engineering from the Technological University of Panama in 2006. Then he got his master's in materials sciences and engineering and PhD in bioengineering in 2012 from the Georgia Institute of Technology. His research focuses on the role of nanostructure and electrical properties of biomaterials in cell differentiation process for tissue regeneration. His work has resulted in one patent and another active application for surface nanomodification of titanium implants. He has published several publications in a highly impact factor journals, co-authored in a different book chapters, and he has also received the American Innovator Young Award. So currently, Dr. Gittens is working on the biomaterial and stem cells for regenerative engineering, as well as applications of mass spectrometry for innovations of public health. With this brief introduction, I invite Dr. Rolando to present his work. Dr. Rolando, please.
Thank you. Thank you very much for that kind presentation and for the opportunity for Dr. Sambasiva's invitation to participate in this very prominent webinar series and for all the organization. It is a pleasure to share with you a little bit of the work that I do here in Panama. It's a great opportunity to talk about my research in a time of a global pandemic, where in a country like Panama, where we're relatively a very small country, especially compared to India, with a very small scientific community, it almost feels like most of our scientists have had to be involved in one way or another to the COVID-19 response and contributing in different ways. So talking again about biomaterials and stem cells actually feels really nice right now. So please just confirm that you're seeing my presentation and that my slide now is on IndicaSat AIP. But basically, I just wanted to give a very brief introduction about what IndicaSat is. I don't know if probably not that many of you have been to Panama or know about our institute, but if you haven't, you should talk to Dr. Sambasiva to fix that, because he has been very involved with IndicaSat, and Dr. Jagannatha Rao, who is our director, establishing the first PhD program in the country. But Panama is a country that is most known for its coffee, also for its sports, like very famous boxer Roberto Duran, for its very booming and growing city, and of course for our biodiversity, in terms of plants, animals, and microorganisms, and also very famous for the Panama Canal. And interestingly enough, our institute is right in front of one of the Panama Canal locks in what is known as City of Knowledge. And within City of Knowledge, we have a building that is relatively new, that is one of the first buildings that has been designed and constructed to do, advanced research. And so IndicaSat has focused mainly on three areas, which are biodiversity, biotechnology, and biomedicine. And as the institute is growing, we're adding new areas of research into the institute, and I'm one of those. I can't say I'm new in the institute now. I joined in 2013, but my area is definitely one of the recent additions to the research areas in the institute, which is bioengineering and tissue engineering in general. So what I'm going to talk to you about today is, biomaterials and stem cells, and specifically to tell you a little bit about some of the results, the latest results that we've gotten published in February of this year related to biomaterials for neuronal differentiation. But what's the motivation of our work? Really, it's focused on a problem that is happening worldwide, and it's the aging of the global population. And this is a problem that is hitting particularly hard countries, developing countries like Panama. You know, this is a population pyramid in Panama in 1950. You can see it has a very nice pyramid shape, as it was expected. But the projections show that for 2050, it's not going to look like a pyramid anymore. And what this means is that the population that is above 60 to 70 years old is going to dramatically increase in the next, 20 to 30 years, meaning that the incidence of diseases that are more common in these populations, chronic diseases, is going to be much higher. And so problems like osteoporosis, stroke, and cancer are going to continue to be a big burden on our population. And we have to come up with solutions, because many of these diseases still don't have good or efficient ways of treating them. So we need to continue to study this. And so in my research group, what we do is that we focus on regeneration as a tool to address some of these chronic diseases. And regeneration has been discussed from many, many hundreds of years, or thousands, if you want to say. But basically, there's regeneration being discussed in mythology. And I don't know if how many of you know this painting, but it's about Prometheus, which was one of Zeus' sons. And he was punished by Zeus by being tied to a mountain and sending an eagle to eat his liver for every day for the eternity. And interestingly enough, now it's known that the liver is one of those organs in the body that has some sort of regeneration capacity. So people are arguing that maybe the Greeks already knew something about regeneration. But leaving mythology behind and going into nature, there are clearly several organisms that show remarkable capacity to regenerate. And I don't know how many of you know this little animal, but it's called the axolotl. And it's endemic from Mexico. It's a species of salamander. And interestingly enough, if you cut one of the limbs of this animal, in a matter of days, where there used to be, bone, muscle, skin, all those cells basically start going through a process of de-differentiation, which is this green blob that you see right there. And in a matter of weeks, those cells are basically able to repopulate that limb again and form all the tissues that were there. And that's why it's called regeneration. That's the difference between scar formation and regeneration, that you get all the functions back the way that they were before the injury. This is a very nice model, the axolotl, and it's been studied for hundreds of years. But the interesting thing is that it continues to produce very amazing papers. And this is a paper that came out maybe a year or 2 years ago, showing that basically they were able to take this model and push it to its limit. Usually we use axolotl as a regeneration model, meaning that you use it to show how it can regenerate. But basically what they did here is that they pushed the model so that they would cut a limb more than 5 times, and they showed that after the 5th time, the axolotl was not able to regenerate. And so it became a very interesting experiment to test why was this model not regenerating anymore. And what they saw is that basically several molecules or markers of scar formation were being upregulated after you pass that 5th injury. And so again, it's interesting to see how the environment, not just the cells, because in theory they're the same cells that are there, but the environment and the extracellular matrix play a big role in the regeneration process. So I'm just going to show this slide because my title talks about stem cells, but this is what I'm not going to talk about in this presentation. I'm just going to briefly, very briefly, talk about how, and something that you probably already know, that there are different types of stem cells. And we have, of course, the embryonic stem cells, which were probably one of the first type of stem cells that were evaluated because they have the capacity to form all the different tissues in the body. And basically, if you take cells from the blastocyst and put them on a petri dish, you're able to differentiate them into all the different lineages of differentiation. In addition, you have what's called adult stem cells. And let me just say, embryonic stem cells, the major issue, even though they're the real pluripotent stem cells in our bodies or in our development, they carry a lot of ethical concerns and ethical issues that have hindered the progress of research using these type of cells. But there's of course, also very interesting discoveries that have looked at adult stem cells, and basically all our adult bodies in our different tissues have adult stem cells, quote unquote, right? In the past, they've been called different things. They're probably one of the first ones that were discovered or that were used and that are currently used clinically are the hematopoietic stem cells, the ones that are in our blood and basically produce most of the blood cells and that are based in the bone marrow. But of course, in many other tissues, they've been able to find some type of specialized progenitor cell that is able to proliferate and repopulate that tissue, either for normal wear and tear or due to injury. Now, in the recent years, maybe in the last 5 to 10 years, there's been some very interesting discussions about how what we used to call mesenchymal stem cells, or this idea of adult stem cell, may not be so general. And there may be no mesenchymal stem cell, but basically each tissue may have a specific progenitor cell that is specialized to repopulate that particular type of tissue. And so it's a very interesting and ongoing discussion right now about what really defines an adult stem cell. And finally, something that has been discussed probably from the early 2000s is the induced pluripotent stem cells, which is a very interesting approach of using adult cells that have been their hard drive erased, through a biochemical bath of factors. And basically you're able to turn the time back on these cells and turn them into stem cells that can become any of the lineages. And initially they were thought of as a therapy that could be used for stem cell treatment in humans. But more and more, they actually have been found to be very, very helpful to develop disease models, because you are basically able to create lab on a chip or organs on a chip, basically very in vitro models of organoids that allow you to test new drugs from the cells that come directly from the patient and give you very accurate information about susceptibility and the reaction that the patient could have to some of these drugs. So they're also a very versatile area of research right now. But what my group studies or focuses on is on what's being called now regenerative engineering, and mainly focused on the biomaterial, which is, amazingly enough, even though it's been studied, the area of biomaterials have been growing since the 70s and 80s as part of the tissue engineering field. But it's still considered not mainstream regenerative medicine. And so what we do is basically we try to develop and study novel biomaterials for regeneration. And of course, that means characterizing all the surface properties and the bulk properties of the material and making sure that it's compatible to the biological application that it's going to be used for. But we can't just create a material and leave it as it is. Of course, we also try to develop in vitro models that allow us to evaluate if this material can actually do what we think it should be doing inside the body. And for that, then we use different types of cells and evaluate how these cells interact with the different properties of the material, especially the surface properties. And then finally, we also use bioinformatics and imaging techniques to try to characterize the regeneration processes. So what I'm going to do now is that I'm going to take you through my experience doing research on bone, and I'm going to use that as an example of the logic and the methodology that we're using to do something similar and developing new biomaterials, but in this case for the brain. And that's where I'm going to introduce some of the results that we've been discussing and that we recently published for neuronal differentiation. So making sure that everything is working fine. Going into bone is a very interesting tissue because it's one of those tissues that actually throughout the lifespan of a person, it shows regeneration capacity. So our bones are constantly going through a process that is called bone remodeling, where we have bone degradation through regular wear and tear or through, injuries and falls and small microfractures that happen inside the bone. But then we have specialized cells that their job is to degrade the bone, which are called osteoclasts, to try to take care and fix those problems that are happening in the bone. So basically, bone is a very complex hierarchical material, if we look at it as a material. And that's why we have so many different bones with different shapes, because depending on the function of the bone, the shape of the bone at the macro scale level is important. But then also at the micro scale level, the complexity of bone is very important, and it's related to the interactions at the cellular level. So like I was saying, we have this type of cell called osteoclast, which goes to the areas, specific areas in the bone that have gone through wear and tear or microfractures, and degrades the bone to clear it up from that problem. And what it does is that it acidifies that area. And interestingly enough, it doesn't have some of the enzymes necessary to remove the proteins, the collagen that is on the bone. So basically, it leaves this very complex nanostructure on the surface of those resorption lacunae. And you can think of it as a combination of micro-sized craters that have very complex nanostructure at the bottom of it. Interestingly enough, it's exactly in those areas where this other type of cell called osteoblast goes and produces the new bone, exactly in that area. And so a lot of hypotheses and that have been proven to a certain extent with many published studies in the literature show that actually it's the combination of biochemical and structural information left by the osteoclast that allows the osteoblast to recognize those areas and produce new bone. So a lot of the studies that I did for my PhD studies and that I continue to work on actually took that hypothesis and tried to evaluate what's really the role of the structure of bone, and in this particular case of titanium implants that are trying to do something similar, that are trying to replace bone, so that we can try to improve the production of new bone around the implant. And so you can think of it schematically as designing implants that have macro scale structure that can interact at the tissue level. But then also it's required for the implant to have micro scale structure or roughness that can interact at the level of the cell. And of course, we also have to design the implant to have nanoscale roughness so that it can interact at the level of the proteins that are being absorbed by this implant at the surface, and that directly and indirectly interact with the proteins that are at the cell membrane of the cells that are interacting with the surface of the implant. So to do this, we basically develop novel nano modification technology to change the structure of titanium implants. And you can think of it as surfaces of implants that come out of the machining process looking like this, relatively smooth. And then after our nano modification process, they will look something like this. So we basically try to replicate or mimic the collagen tufts that I showed you before that are on the surface of those resorption lacunae, to try to mimic that hierarchical structure on the surface. We were able to show that these techniques were able to be applied on complex 3D structured implants like spine implants, titanium spine implants for orthopedic surgery. And we also evaluated, of course, the biological response on these surfaces and showed that actually the cells are able to interact with these nanostructures that are being created on the surface. And of course, we quantified this, evaluating the proteins and the gene expression of these cells growing on these surfaces. So the main point that I want to share with you from this experience in bone is that we clearly were able to show that the surface properties of the material where the cells are growing has a big influence on the way that the cells behave, on the proteins that they produce, on the gene expression that is going on inside the cell, And so all of that is basically a reflection of how cells need to interact with their environment and the extracellular matrix to basically understand where they are and what they need to do. And so what we're doing is that if we mimic the properties, the natural properties of bone, and put that on the surface of a titanium implant, then we're telling the cell, you're in your environment that you enjoy, so you can start producing proteins necessary for bone formation as fast as possible so that the patient can heal faster. So that was a great lesson that we learned working on titanium implants for bone applications. And so I've always have had a fascination with brain and the way that brain cells behave. And I didn't mention it before, but the background of my presentation is one of these beautiful Ramon y Cajal slides and drawings that he would do of the neurons that he was able to visualize. And the most amazing thing that I've always noticed in these beautiful drawings that he made is how he focuses on the cells, but these cells are almost like floating in space, right? Like there's nothing in between them holding them in place. And in reality, we know that there's a lot of extracellular matrix doing that job, even in the brain. And so from my experience with bone and titanium implants, the question then was how the neurons interact with their extracellular matrix and with biomaterials that we can design to treat problems in the brain. And in this particular case, we're talking about stroke. Stroke is a problem that can happen either through ischemia or through a hemorrhagic problem, where the one that we're presenting here, it's a stroke caused by ischemia, where there's a blockage of some of the blood vessels in the brain that cause that an area of the brain stops receiving oxygen and nutrients. And the brain clearly and neurons are very energy intensive cells that as soon as they're cut off from their source of oxygen and nutrients, start suffering very quickly and even dying. So the problem then becomes, once you have that stroke, if you don't take that patient quickly to the hospital, within what's called the acute phase, you go then into a chronic phase where the neurons that are in what's called these area of the nuclei of affectation then start dying. And that's one of the biggest problems of patients that go into that chronic phase, because it definitely makes it harder for come back from that cognitive and motor impairment that patients that suffer stroke go through. And so the idea that we're proposing is, well, right now there are really very few options available for patients that go into that chronic phase. If you go into the acute phase and you are able to go into the hospital and they're able to remove that insult, that ischemic insult, then the probabilities of recovering your cognitive and motor abilities is relatively high. But once you go into that chronic phase, the options are very limited. So the question is, can we design a technology, a therapy based on biomaterials and stem cells that can be applied in the area of the insult and that can help at least stop the chronic neuronal death that happens in that region, and in the best case scenario, try to revert some of that damage through regeneration. And the idea is approaching it in a similar way, where we're designing these biomaterials at the microscale level so that they can interact at the level of the cell, but also at the nanoscale level so that they can interact at the level of the proteins that are in the surface of the biomaterial and in the cell membrane of the cells in the brain. So, of course, brain is not bone. Brain is a lot more complicated than bone. And just to show you an example, this is a figure from one of the papers from the group of Herculano et al. From Brazil, where they've done this amazing work trying to characterize the differences in cell content in the different brain regions. And as you can see here, we think of the brain as a whole, and most studies that look at brain extracellular matrix look at the brain as a whole. And we think that, well, brain is just tissue and cells, and you can think of the cells in the brain as being either neurons which are about 86 billion neurons, or non-neurons, which includes microglia, oligodendrocytes, and all these different other types of cells that you can find in the brain. And it's about 84 billion of those non-neurons. That's in the whole brain. And it doesn't seem to be that different, right, between one or the other. But once you start looking at particular subregions of the brain, for example, the cerebral cortex, which is 81% of the brain mass, it only contains about 19% of the brain neurons. That means in the cortex, you only find about 16 billion neurons and 60 billion of the non-neurons. So most of the non-neurons are in the cortex, but not the neurons. On the other hand, you see a region like the cerebellum that has about 69 billion neurons. So most of the neurons in the brain are in the cerebellum, but only about 16 billion non-neurons. And the cerebellum is only about 10% of the brain mass. So all these differences in mass and in neuron content and in non-neuron content is very important to understand from the perspective of the extracellular matrix, because clearly the extracellular matrix is going to be tailored for the type of cells that you have in that region. So if you get a stroke lesion or injury in the cortex, then you want to make sure that the extracellular matrix that you're using to treat that is also composed of similar extracellular matrix as you would find in the cortex. And so that's something that we need to consider. The other thing that we need to consider is this interesting discussion that is happening in these last few years, where in the field of stem cells for the longest time, the idea was that if you had stem cells and injected them in an area of injury, those stem cells would then go and interact with that injury, start differentiating themselves, and then mature and become the actual tissue that you wanted to repair. But more and more studies are coming out for many, many years now, having very much difficulties trying to prove that that process is actually happening. And now showing that probably the biggest effect that cells have on the injury side is actually through paracrine factors. So these paracrine factors are probably being produced by the cells that are being injected. They're being released and have an effect recruiting stem cells or progenitor cells from that tissue, so that then those progenitor cells are actually the ones that are becoming and repairing the tissue. And these cells that you injected that are, probably not necessarily from the patient, are gonna be removed in a matter of hours or days. So these two processes are probably happening at the same time, but more and more data and evidence is showing that the paracrine factors are actually maybe playing a bigger role in the regeneration process. And this graph came out in a review paper that we published in 2017 in collaboration with Dr. Miguel Perez Pinzon from University of Florida. So now I'm gonna go a little bit into the study that just recently was published by our group, where we basically take this concept and apply it to brain extracellular matrix, where what happens or how does the different brain subregions behave when they're decellularized? Are they all able to differentiate stem cells, or in this particular case, PC12 cells, a unidirectional differentiation model, in the same way? What I mean with this is, if we expose these cells to decellularized extracellular matrix from the cortex, do they behave the same as if they were treated with decellularized extracellular matrix from the cerebellum or from the remaining area? So that's basically what we're trying to show here, where we took these three different regions from the brain, decellularized that extracellular matrix, removing all the cellular component, and using this as a biomaterial to treat unstimulated PC12 cells and see which one was better at differentiating these cells. And let me just... So going back at this idea of brain complexity, it was important to us to also show what the team from Herculano et al. Showed. And basically, we were working with porcine brains. Actually, at the time, there was very little information about the cellular content of these different brain subregions from pig brains. And so what we showed was that basically evaluating, using the same methodology, evaluating the weight of these different brain regions, again, cortex, cerebellum, and remaining areas, and looking at the number of cells in each one, we show basically that the cortex has about 25% of the cells in the brain versus the cerebellum, which has about 55% of the cells on the brain, and the remaining areas have about 20% of those cells. So interestingly enough, for our purposes, which is to decellularize this brain subregion, we find that the cerebellum probably will be the most difficult area of the brain to decellularize because of its high content, its high cellular content. So that was very valuable information for us at the beginning to try to understand how these tissues were going to behave during our decellularization process. And let me just clarify that we're not the first team to work on deceleration protocols. I mean, deceleration of extracellular matrix has been going on since probably the 80s. And initially was used for porcine intestinal submucosa, probably one of the first tissues that was decellularized on purpose. Before that, clinically, tissues like the amniotic membrane were being used to treat, injuries in the eyes and in other areas of the body. And what many of these studies have been able to show, some of these products are actually commercially available products, showing that they're capable of promoting tissue regeneration in different parts of the body. So again, we're not the first team trying to show decellularization and how the decellularized tissue can help stem cell differentiation. But in particular, what our approach was trying to show was how brain, because of its complexity and the different subregions of the brain, cannot be treated as a single organ or as a single tissue for decellularization. And so to do this, we took available protocols on the literature, one that was done over 4 days using a very simple reagent based on SDS, a detergent, to remove the cellular components throughout the 4 days. And another protocol that was developed later, that was a 1-day protocol, that was a little bit harsher in terms of the reagents that were used, including trypsin, tritonex, sucrose, and other, peracetic acid, but done over a period of less than a day. And to try to see, and both studies or both protocols in the literature had shown that they were capable of decellularizing brain tissue. But again, brain tissue without specifying from what region of the brain they were working with. So what we were able to show, at least, is if we treated the different brain regions similarly, and we basically loaded 15 mL in a conical tube of these different brain subregions and process them with these different protocols, the end process or the end result was very, very different. So basically, tissues like cerebellum were consistently lost at higher degree compared to cortex or the remaining area. And we were able to evaluate that in terms of the weight of the tissue and the volume that was clearly visible in the tube. And we think that that's possibly because, again, what we're trying to do is remove the cellular component of these tissues. And like I told you before, the cerebellum seems to have the highest percentage of cells from all these 3 different brain subregions. Visually and graphically, what this looks like is that basically, again, we would dissect these 3 different brain sections from the porcine brains that we were working with. And after passing them through the decellularization process, they would look something like this. So they would lose all their coloration, and they would significantly decrease in volume. And then after we had a tissue like this, we would lyophilize it and use it to test it for microbiological analysis, just as a quality control to make sure that we were able to work with biomaterial that was sterile, or at least, aseptic. And so a very important part of the study was to characterize these decellularized extracellular matrix, again, because we wanted to tell or know what were the difference in these different subregions. And so there's a lot of information on this graph, but let me see if I can take some time to explain it. But basically, one of the most common techniques to prove that you're actually being capable of decellularizing a tissue is quantifying the DNA that it's inside that extracellular matrix that you're decellularizing. The idea being that it's an indirect way of evaluating if there are still cells in the tissue or not, right? So if you have decellularized a tissue, but you still find DNA levels inside that tissue, in an indirect way, you can say that, well, basically, you were not able to remove all the cells that were on that tissue. And of course, it's understood that you're not going to be able to remove all the DNA, even if you remove all the cells, because in the process of them breaking apart, there could be DNA that is left in the extracellular matrix. So the gold standard is that a good deceleration protocol should take you down to 100 nanograms of DNA per milligram of dry tissue. And so what we have here in panels, sorry for the letters not being in the right order, but in panels C, D, and E is the 4-day protocol for cortex, cerebellum, and remaining areas. And as you can see here, at day 0 before decellularization, the DNA content in cortex is about 1,000 nanograms per milligram. In remaining areas is about 750 nanograms. But in the cerebellum, we're talking about 2,000 nanograms per milligram of dry tissue. Coinciding with what we found, that indeed the cerebellum is the one that has the highest cellular content. And what we saw throughout the protocol with the 3 different subregions was that immediately after the first day of decellularization, there was a significant decrease in the content of DNA in all the tissues. So you can see here right away a significant decrease in the levels of DNA. And this decrease continued until day 4, where we found the lowest level of DNA content in each one of these tissues. In the 3 cases, we were able to reach that gold standard of being below the 100 nanograms per milligram of dry tissue. And when we evaluated the other protocol, the 1-day protocol, using the harsher reagents, we were able to see a similar picture. Basically, you can see here cortex, cerebellum, and remaining areas. The black bars are the native tissue, so tissue before decellularization, and you can see how the cerebellum is the one that has the highest content. And then as soon as we're able to decellularize this tissue with that protocol, we can basically remove most of the DNA content in those tissues, again, proving that this protocol is capable of decellularizing brain tissue. Now, let me just mention that it's clear that brain decellularization is not as popular, and you can't find that many studies in the literature showing decellularization of brain tissue because it's a very difficult tissue to work with. Through the process, you lose a lot of tissue, and you have to be very, very careful with handling the tissue so that you can salvage and maintain the decellularized extracellular matrix while you're processing it. So we also, confirm these results of DNA quantification through other techniques, basically using histology, basic H&E staining, where you can see the DNA the DNA in the nucleus of the cells in purple and the rest of the tissue in pink. And you can see here on the top row, the native tissue, again, cerebellum showing a higher concentration of nuclei than the rest. And then on the bottom row, you can see the decellularized tissue with much less content of DNA, or at least of nuclei. And you can see also some gaps, of course, some of the tissue that is lost during the process. But we basically show that, again, cerebellum, native cerebellum has the highest level of nuclei concentration. And after deceleration, the three areas are able to significantly decrease in content. And one more time, we show the similar thing with DAPI. And basically, you can see here how after deceleration, most of the nuclei have disappeared. Another aspect that was important for us to characterize was the actual extracellular matrix structure. So you can see here, again, we have the native tissue on the top row. We have the decellularized tissue with the 4-day protocol on the middle row, and the decellularized tissue with the 1-day protocol on the bottom row. And the important things here to show are that, again, with both protocols, we see a much lower presence of these nuclei in the tissue. And we can see that there's a significant loss of tissue with the 4-day protocol compared to the 1-day protocol. And this is what we're quantifying here. Basically, we graphed it as tissue preservation. And what we're doing is that we're quantifying the gaps in the tissue. And you can see basically how for the 4-day protocol, there's basically less tissue preservation, meaning bigger gaps compared to the 1-day protocol, which is very similar to the native tissue, okay? Another important aspect of tissue decellularization, in this case, extracellular matrix, decellularized extracellular matrix from the brain, is what are the growth factors that are available in the extracellular matrix to promote stem cell differentiation once we use it as a biomaterial. Because clearly, I've told you already that structure is important for promoting stem cell differentiation, but biochemistry is also very important. And for the particular in vitro model that we're using, which is a PC12 cell line, we know that this cell line responds to NGF and to BDNF, two very important nerve growth factors or neurotrophic factors. So what we quantified here is the protein concentration. And as you can see here, we show that we're able to maintain most of the protein after decellularization in the cerebellum. It's a little bit more complicated, as I mentioned before, probably because of the high loss of cellular content. But we're able to maintain enough protein content from these tissues. But once we start evaluating the presence of specific growth factors, in this particular case, NGF, we show that after decellularization, it doesn't matter if it's a 1-day protocol or the 4-day protocol, we basically lose all presence or quantifiable levels of NGF. Interestingly enough, we're able to find that the levels of BDNF, brain-derived nerve growth factor or neurotrophic growth factor, actually increases as the deceleration protocol is applied, particularly in the 1-day protocol for the remaining areas. That doesn't, it was really hard for us to explain how could it be that a growth factor was actually increasing after decellularization. But the explanation that we gave, and we're still, of course, evaluating this, but is that what's happening here is that this is normalized by the dry weight of the tissue. So what we think is happening is that when we are decellularizing the tissue, we're removing certain components of the tissue that are not necessarily proteins or BDNF in this particular case. BDNF is known as a growth factor that is associated to the extracellular matrix through extracellular vesicle. And so we think that because we're losing some tissue, but maybe not the tissue where the BDNF is the BDNF is binding to, we think that it's possible that we're enriching the tissue with BDNF by maintaining it in the extracellular matrix that we're able to keep. So again, the important thing here is to show that in this particular characterization, we're able to show that the biomaterial that we're working with doesn't have NGF, which is a well-known activator of PC12 cells, but we are able to show that we do maintain some of the BDNF, which is also capable of differentiation PC12 cells. So what are we trying to look for then? In a very simple schematic way, we're doing in vitro culture of PC12 cells. And on one side, we have certain controls of PC12 cells that are not exposed to the biomaterial. And those cells will grow something like this, where they grow in spherical colonies and don't express or show any signs of differentiation, versus cells that are exposed to a maturing or differentiating environment that start producing these axon-like structures, signaling that indeed they are in an environment that is conducive to neuronal maturation. So to do this, we basically applied image analysis techniques where we're able to quantify the extension of axons using ImageJ software. And basically, depending on the length of these axon-like structures, we're able to show cells that have axon-like structures that are larger than X amount, we consider to be cells that are differentiating. And that's how we quantified the amount of differentiation. And so for the publication, what you can see here, again, is our control cells. As you can see, very, very little to no differentiation. These cells have not been exposed to anything other than saline solution. NGF, this is the positive control with NGF. As I mentioned before, it's very well studied and known that NGF will differentiate PC12 cells very quickly and make them produce these axon-like structures. And BDNF can do it as well, just not as effectively as NGF. As you can see here, several of the cells are indeed producing some of these axon-like structures, although smaller, in a smaller extent. And so when we do a similar evaluation on our 4-day protocol or on our 1-day protocol for the 3 different brain regions, cortex, cerebellum, and remaining area, you can see that indeed, first, very importantly, these cells are able to grow and behave when they're exposed to these biomaterials. I'm not showing it here, but of course, we also did some experiments showing the viability, cell viability with our biomaterials. And we show that the biomaterial has basically no effect, no negative effect on cell viability. And so when we try to quantify the differentiation or the cell maturation when exposed to these different biomaterials, what you can see here in this graph is that, as expected, the positive controls have cell maturation percentages of, above 50% for NGF or around 45% with BDNF. So 45% of the cells showed some axon-like structure with these 2 very potent treatments that we tried as positive control. But when we use the biomaterials, we see that indeed they're able to promote cell maturation compared to the negative control, but to a lesser extent than what was seen just using NGF or BDNF. And so what we think is happening here is that the biomaterial by itself has much lower concentrations of these growth factors, as I showed you, probably very close to 0 concentrations of NGF, but very small concentrations of BDNF, but still lower than what we were using as positive controls. And again, we also think that the structure, the interactions of these cells with this biomaterial is also playing a role in this differentiation that we're able to see here. So that was basically the study that we were able to publish. We continue to evaluate different things that we found interesting in these evaluations. For example, we've been trying to do primary cell culture on these biomaterials using cells that are extracted directly from fetal brains of rats. We're able to show that in the control coatings like poly-D-lysine, we see a very homogeneous covering of these cells or growth of these cells. And you can see here the presence of both neurons and glia in the culture. Interestingly enough, when we start growing the same cells in the cortex, the cerebellum, or the remaining area, we see that, again, these cells are able to grow which is already an achievement for us. But the presentation of the cells on the surface is very different. So what we found is that these cells start aggregating in clumps when they're growing in these different biomaterials. And right now we're trying to evaluate if this is just an artifact of the coating with the biomaterial, that maybe we're not being able to produce a homogeneous coating on the surface of the cover slip, which, you know, we don't think that's the case, because we did characterize the process of coating on cover slips, and we show that we do indeed have homogeneous coatings with these biomaterials. But the other idea is that maybe these biomaterials are letting these cells behave in a more natural environment, and this may be a more natural presentation of the interactions between these cells on the surface. So we're still evaluating these aspects, and we don't have any definitive answers for it, but we thought that it was a very interesting finding with these primary cells. So just to finish now, the next steps where we are, you know, what I showed you so far was using a simple model using a PC12 cell line, which is not necessarily a stem cell, because it's just a unidirectional model. It's either undifferentiated or differentiated neuron. So that's a unidirectional model. But we want to use now a multidirectional model using neural stem cells, where these cells in theory can differentiate into different lineages, either astrocytes or oligodendrocytes or neurons. And we want to show that indeed our biomaterials are able to preferentially differentiate these neural stem cells into neurons, to promote this idea that maybe some of these biomaterials can be used to treat a problem like stroke. In the long term, and I just showed you this as a very interesting video to enjoy, but the possibilities with decellularized extracellular matrix are fascinating. This is work done by the Ott Lab in Harvard, where they're basically decellularizing entire organs. And this may not be so applicable for the brain, but they decellularize entire lungs of mice, and then they're able to repopulate these lungs with their own, the mice own stem cells, and show that they're able to recuperate the function of the organ again in vitro, so, or ex vivo at least. So it's just fascinating to see all the different things that are able that can be done with perfecting the techniques of deceleration. So with that, I'm done with the presentation, or what I had prepared for you, and I would be very happy to answer any questions that you may have. And thank you again for the opportunity to present and to share with you some of the work that we're doing here in Panama.
Yeah, thank you, Dr. Rolando. Now I request all the participants to post their question in a Q&A box.
So should I write answer live? Oh, live. Live means that I can talk. Okay. So one question is, what type of brain model you use? And what we used basically was a porcine brain. Can you hear me? Yeah, so we were using a porcine brain, porcine brains that we collect from a local
This
abattoir.
is the power system here for me. I'm living in dark in my room. Yeah. Since 2.5 years, I'm in dark only. I never worked. There is no backup power for me. You ask them.
Could we mute one of the channels that okay.
No, so we have money. We are getting crores of money, but vice chancellor office is in dark.
Sorry. So anyway, yeah, we're using porcine brains that we collect from a local abattoir, and with that, we just take them back to the lab, do the dissection of the 3 different regions, and then store them for future use, or use them right away and start the deceleration process. So the next question, are there studies to show that neuronal regenerating cells to alleviate conditions in dementia and Alzheimer's patients? That's a great question. So right now, even from the 90s, there's been some studies where they've applied stem cells for treating, for example, diseases like Parkinson, that are, again, they're less complex than Alzheimer's in that we know that it's a localized problem in the case of Parkinson's with the substantia nigra and the cells that stop producing dopamine. And they've been able, there's a very interesting study that showed that they were able to inject stem cells in that part of the brain. And they've been able to follow up some of these patients for 10 or more years and show that some of these stem cells are still there. And they did show, at least with some of the patients, that they were able to have interesting results, but not with all. So it was very variable. And I know that there are still many clinical trials going on trying to do that. I think that for Alzheimer's, there's less information available, and probably it's because of the complexity of the disease that affects the entire brain. So it's harder to pinpoint where would you give the treatment to show that indeed stem cells are useful. And so, have you transplanted the tissue engineered scaffolds to the animal model? So we so far have not used our biomaterials in an animal model to show that indeed we can use it to regenerate parts of the brain. It was part of the study, and sorry that I need to drink some water. So this study was financed with a grant from NIH and in collaboration with our colleagues from the University of Florida. I'm sorry, University of Miami. And part of the project does look, is interested to see if we can put these biomaterials in an animal model, in a rat model, to see if we can indeed show some regeneration capacity in the animal. But the next step actually before that will be to do an organotypic model to show that indeed we're able to treat an organotypic model with these biomaterials and show what's the response. And so, any comments on skin regeneration? I'm sorry, I have not worked with skin so far, but I can tell you that skin is probably one of the first commercially available tissue engineered solutions. So there's plenty of work done on skin regeneration using stem cells, but I particularly have not done work on that.
Rolando, this is
yes.
Rolando. This is Sambasura. What about the tissue generation, regeneration in tissue of bones? It is as good as normal tissues, or it is having different type of regeneration?
Well, I think that tissue regeneration on bone has a lot of tradition, and there's been a lot of work going on tissue regeneration around bone, in part because bone, like I said at the beginning, has a lot of capacity or ability to regenerate by itself, right? So for bone regeneration, you can use other type of materials, right? A little bit, harder materials. So we work with metals and ceramics. But there's very interesting technologies available to do bone regeneration. And for example, I think that one of the areas that it's always pushing the boundaries on bone tissue regeneration is the dental field. So actually, dentists are constantly having to augment bone, both in the maxilla and the mandible. And so in there are very interesting advances in tissue regeneration for bone.
And it is when you are having some kind of arthritis problem, people are telling that we can inject stem cells at the place of arthritis and also knee joints. That is also some doctors, clinicians, they are mentioning here that they can they can cure the problem of arthritis by the way of stem cells treatment. What is your opinion on that?
Well, it's very interesting, right? Like there's been this whole push to try to show results with stem cells in clinical applications, in different clinical applications. I think that osteoarthritis is probably one of the most sought after treatments with stem cells. There are a lot of anecdotal results saying that it works, and there's a few studies published showing that indeed with a single injection of stem cells, you can have improvements in pain and mobility of patients for up to a year. But it's been hard to replicate because there's not enough studies showing that. And so far, there's still the only stem cell approved treatment, I would say worldwide, but based on the FDA regulations, it's still only stem cell transplantation through bone marrow transplantation, right? So hematopoietic stem cells, but not for treatments of osteoarthritis of others.
That bone marrow is from the autologous patients or allogenic patients?
You can use both, actually. You can use both. Usually, actually, it's from another patient, right? So because what you're trying to do is to repair, in cases of leukemia or leukemia, trying to replace the blood cells for the patient because he has cancer, right? So you have to use a validated donor that meets the hematological parameters to be able to use them. But with that, I don't want to say that other stem cell treatments don't work. I think that they work, but we haven't been able to show that they work consistently. And I think that part of it is because of the complexity, one, of the method of delivery. So a lot of the treatments that are done currently in clinics that are doing stem cell treatments are done intravenously. And that kind of dilutes a lot the effect. And part of it is that the effect is an immunological response, right? So the stem cells are being able to regulate the inflammatory response in the person. And so there's a lot of variability with that as well.
Yes, it is good actually. And you can carry on with other questions. No, I'm just, if I get another doubt, I will ask you. Okay.
Of course.
Thank you.
So let me see. There's another question here about, like, if few virus attacks, for example, bacterial meningitis, blood-brain barrier is mainly affected and the virus penetrating into it, what are the chances of regenerating it? Not sure I can answer that question. I think that it's hard to know. And I don't know if studies have looked into regeneration after virus infections, but it would be actually very interesting to know right now, right? And actually, we have a colleague here in Panama that is doing some research on stem cell treatments for COVID-19 patients. This would not be for brain regeneration. It's initially focused on lung regeneration, because we know that COVID-19 actually affects the lungs of the patients, among other tissues. But yeah, I think that at the end, if the injury, independently of what the source of the injury is, if the injury ends up affecting or killing cells within the tissue, the idea is that through stem cells, you can try to regenerate that damage. So another question is, can we co-culture all stem cells to produce inner mass of cells in blastocyst stage by using nanotechnology or biomaterials? That's a great question. And I think that there's some work done in China, actually, where they're using stem cells to create in vitro fertilized eggs that then they can use to create embryonic stem cells. And I guess it's a roundabout way to avoid the ethical concerns of embryonic stem cell research, but it's still problematic. So it's harder to do work related to blastocysts just because of the ethical concerns. Another question is, do biomaterials trigger immune responses? And that's a great question. They definitely do. And I think that that's where a lot of the research needs to focus on. What kind of immune response your biomaterial is going to produce? Because what's been shown with extracellular matrix, decellularized extracellular matrix, is that if it's done correctly, meaning if you're able to remove the cellular component and bring the DNA content under the 100 nanograms per milligram of dry tissue, then the immune response that you're able to produce in the host is actually an anti-inflammatory immune response that leads to regeneration. And they think actually that it's because of that immune response that you're promoting regeneration. But if your biomaterial is not treated correctly and contains higher content of cellular debris or if it's being cross-linked instead of just being the regularly loose, extracellular matrix, then you can cause the opposite effect, which is an pro-inflammatory immune response. And that actually can be very problematic, because then what you're causing is that you're causing more damage to the tissue, and it's what you're trying to avoid. So another question is, have you ever tried kidneys or liver that have specialized cells that are affected and cause high social impact disease? Wonderful question. I personally have not worked with any of these tissues before. Actually, I was trying to do a collaboration with a laboratory, a hematology laboratory in one of our main public hospitals that have a wonderful certified laboratory to do bone marrow transplants, and they also have done other organ transplants. So they do kidney transplants as well. And they're very interested to know how we could apply some of these techniques and technologies of extracellular matrix deceleration and stem cells to treat some of these patients. But we haven't started yet. So I don't have results for that question yet, unfortunately. Do biomaterials trigger immune response? I already answered that one. In your work, you use NGF growth factors. Does these growth factors show good progress? Did you try any other growth factors? We mainly focused on NGF and BDNF because of the cells that we were using in our in vitro model, which were the PC12 cells, and they are shown to respond to these factors. But yes, indeed, we're very interested to know what other growth factors are in the extracellular matrix that could also be playing a role. But right now, I don't have any results to show on that aspect. Can the regenerated nerve tissue regain the functionality too? That's also a great question. I personally have not worked with nerve regeneration. My colleague that was leading these studies in the lab, called Diego Reginensi, actually has specialized in nerve regeneration. And I think that it's difficult, but it's been shown that indeed, especially through using Schwann cells, you're able to promote some of the nerve regeneration as well. Can you give some brief on cell mutation process? I'm sorry, I cannot contribute on that.
What is your opinion about scaffold development in for various tissues? Scaffold development.
On what tissues? Sorry, Dr. Sambasio.
Like maybe kidney or liver, or regeneration in the laboratory conditions. No scaffolds. That is regeneration of the tissues in the laboratory. We are developing the models in the form of scaffolds, and regeneration of the particular organs, organ development. What is your opinion on that?
I mean, I personally think that scaffolds are very important, and I'm still very surprised that it's not mainstream, that many, even clinical trials with stem cells are not using biomaterials or scaffolds to promote the differentiation and the regeneration that they're looking for. I think that personally, clearly I'm biased because I specialize on biomaterials, but I think that the regeneration story has two components. One component is stem cells, and that's what we all hear about and know about. But the other very important component, almost just as important as the stem cells, is the biomaterial and the scaffold that you're using as a vehicle to deliver those stem cells. So I think that scaffolds are very important for many processes. It could be for the treatment, but it could also be for the preservation of the cells. There are many studies showing that actually.
What is the possibility for tissue regeneration, organ regeneration, particularly liver damage, kidney damage? Are we able to have success in this particular situation?
I think that we're getting closer and closer, especially with some of these groups that are working with full organ decellularization. There are actually some companies already in the United States that are doing liver decellularization to try to repopulate that extracellular matrix with the patient that requires the organ transplant, grow them in the organ, and then you do the transplant. So I think that there's been great progress in that perspective. And so I think that we have to continue to work on understanding what are those key properties from the scaffold or the biomaterial that we need to maintain so that these stem cells can survive and do the work that they need to do.
Okay, thank you. Thank you, Ricardo.
Of course. I think I've answered most of the questions. I mean, there are some others. Are stem cell treatment safe? I think that that really depends on the clinic and the regulations that are being preserved in each country.
Thank you very much for your kind help.
Of course. No, thank you. Thank you for the wonderful questions and the opportunity again to participate in this very special
very good.
occasion.
No, very nice information to all the participants. Are very much appreciating your lecture and presentation and information. And I saw many appreciations from participants. Around 190 to 200 people attended the session, and almost everyone is appreciating your lecture.
That's amazing. No, thank you very much. And I send you many, many salutations from Dr. Rao. And you know,
Yes,
missing the times when you visit us in Panama.
Yes, yes. I'll look for the opportunity to visit Panama again. Yes, thank you. I think over to our people, Amit Trivedi will take over.
Yeah,
I will take leave. I will take leave. Thank you. Thank you. I will see you again. Thank you.
Thank you.
So, Dr. Rolando, I think still there are a few questions left. If you can answer those questions.
Of course. Let me see. There's, what was the type of deceleration that you have used? That in particular for this study, we did 2 protocols. We did a 4-day protocol that was based on SDS detergent, and then a 1-day protocol that was based on harsher reagents. It's a more complicated protocol because we use maybe 5 or 6 different reagents to try to remove the cells from the matrix, but just through 1 day. And what we show is basically that the 1-day protocol is just as effective as the 4-day protocol, but because there's less mechanical forces involved, it's just over 1 day, it maintains the extracellular matrix structure better. So I would recommend the 1-day protocol for sure. And then would it be a good guess to say cytokines or interleukins promote tissue regeneration? And I think that it's hard to answer that question. They will definitely be involved in the process. As I said, you know, a big component of tissue regeneration is the immune response. And the immune response is based on cytokines and interleukin production. So it's well known that some of these factors play an important role, but it's not just one factor, right? It's a combination of events that have to happen so that you have the regeneration response that you're looking for. So by using biomaterials, can we regenerate heart cells? That's a wonderful question. I think that there have been many, many studies trying to show heart regeneration using stem cells. And so far, we have not been lucky enough to find a solution for that problem. There's been even studies published showing that there's very few or very little hope left to show that indeed you can regenerate heart cells using stem cells. But I think that it's one of these things where, as hard as it may be, if we continue studying and evaluating possibilities, then we may find a way to do it. But so far, we have not been lucky with heart regeneration. To overcome immune response of biomaterials, can brain of dead human body will be used to make biomaterials? Well, I mean, in general, that's a great question, in that one of the things that we're showing is that if you're able to use decellularized extracellular matrix from the same tissue that you're trying to regenerate, right? Because right now, some of the commercial products are intestinal submucosa or amniotic membrane, and that extracellular matrix is being used to treat osteoarthritis, injuries in the eye, muscle injuries. Clearly, there's a discrepancy between the extracellular matrix that you find in the amniotic membrane and the extracellular matrix that you find in muscle. But even though there's that discrepancy, you're still able to have a good effect, probably because of the growth factors and the cytokines that are available in that decellularized extracellular matrix. So what I'm trying to say is that that also applies to the species of the source of the tissue that you're collecting. So the ideal would be to work with human tissues. We know that probably it will be a lot difficult to get enough material from human source to be able to produce it as a solution or as a medical treatment. But I believe that it probably would have a better response. Are you differentiating any other neuronal cells like astrocytes or oligodendrocytes in vitro culture? Well, not so far, but we're very interested to get there. And some of our studies now are looking into that multi-directional model that would allow us to evaluate some of this. And then would it be a good guess to say cytokine or I already answered this one. And then can we store the neuronal memory during tissue culture or regeneration? That's a wonderful philosophical question. And I don't have an answer for that question. I mean, I think it's a great question that needs to be discussed, especially I think that the approach that we're trying to use of just using a very localized application of brain extracellular matrix in one particular area of the brain suggests that we're not really affecting memories or the personality of the person that is receiving the treatment. Now, when we're talking about full organ regeneration, that's why I think that for the brain, it may be a lot more complicated to consider an application of that sort. So I think that it's an important consideration, and I would be very interested to explore ways of evaluating that while we're doing decellularization of the tissue. But I think that it would lead to a very interesting paper.
Yeah, Dr. So there is one question in the chat box, and that question is, have human embryonic stem cells been used successfully to treat any human diseases yet?
That's a great question. I think there have been applications of embryonic stem cells in humans. There's actually a company, at least one or two, that actually produce embryonic stem cells for human treatment. And I think that for actual medical treatment, I don't have a particular application I can share with you right now, but I think that it's actually from the research perspective that embryonic stem cells have provided many, many important insights and that justify the need to be able to access those tissues. And I think that if we're able to do it in an ethical manner, fulfilling all the regulations that every country can set for this type of research, then I think that it's valuable research that should be done to try to understand better how our body behaves and how our different tissues behave.
Okay, so I think with this, we have come to the end of this session. So again, I would like to thank Professor Chiara Sambasiva Rao for initiating this webinar series, which is allowing us to interact with many foreign faculties. And I would especially like to thank Dr. Rolando for sparing his time and delivering an excellent lecture. And I will also thank all the participants for enthusiastically participating in this webinar and ask the several questions. At the end, I will also thank our ICT persons, especially Mr. Suraj, because today in very extreme conditions, we have a heavy rain since yesterday night, but still he managed all the things and successfully we have come to the end of this program. With this, I thank all of you. Thank you.
Thank you, Rolando.
Thank you very much. Thank you.